This is glycolysis. A synonym of this is EMP or Embden-Meyerhof pathway. Glycolysis is the breakdown of glucose to pyruvate in aerobic conditions or lactate in anaerobic conditions for energy. And the sites in tissues, it occurs in all the tissues, and this is the only pathway that occurs in all the cells of the body. Intracellularly, it occurs in cytosol outside mitochondria. It starts with glucose, and end products are pyruvate in aerobic conditions and lactate in anaerobic conditions. Conditions for anaerobic glycolysis. Anaerobic glycolysis occurs in hypoxic tissues, vigorously contracting muscles, skeletal muscles, in RBC, lens, etc. These lack mitochondria, so the reduced NAD cannot be used, cannot be oxidized in mitochondria since there is no mitochondria. Lactate is formed so that NAD can be again oxidized from NADH plus H plus. The reaction pathway of glycolysis. In the first step, glucose becomes glucose 6-phosphate with the help of the enzyme hexokinase and glucokinase only in liver. Hexokinase in other places except liver, and ATP is used with the help of magnesium ion. ATP is used as coenzyme for this enzyme. In the second step, with the help of phosphohexose isomerase, this glucose 6-phosphate becomes fructose 6-phosphate. Okay, isomerase enzyme changes the structure, and there is another similar enzyme mutase which transfers the functional group from one position to another position. In place of phosphate group, I have given this P, encircled P. In the third step, this fructose 6-phosphate becomes fructose 1,6-bisphosphate with this enzyme PFK, phosphofructokinase, and using ATP. And here this step is actually reversible. Okay, step number 2. This is the first step, this is second, this is the third, and this third step is rate limiting step, RLS. And this is the fourth step where this fructose 1,6-bisphosphate is converted to glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. These are 3 carbon compounds, and this is 6 carbon compound. These are phosphate groups, and here is some changes in the double bond. Here, this reaction is reversible, and the enzyme is phosphotriose isomerase. Again, isomerase, change in structure. And this is the fifth step where we actually get 2 molecules of glyceraldehyde 3-phosphate because this is interconvertible. And with the help of NAD plus and inorganic phosphate, this glyceraldehyde 3-phosphate is converted to 1,3-bisphosphoglycerate. 2 molecules of this, and 2 molecules of NADH plus H plus is formed, which is equivalent to ATP energy when the Krebs cycle is operating. The enzyme used is glyceraldehyde 3-phosphate dehydrogenase. And the next one, this is step number 6, where 1,3-BPG is converted to 3-PG or 3-phosphoglycerate. This is actually 2 molecules. Yes, 2 molecules, and here directly the energy is formed ADP to ATP using Mg2 plus. Yes, this enzyme is actually named for the reverse reaction when the phosphoglycerate is kinased by kinase enzyme. This forms. This is actually for reverse reaction, and this reaction is actually reversible. This is actually reversible reaction, and this one was also reversible reaction. Then step number 7. Step number 7 is also reversible, and this is catalyzed by the enzyme phosphoglycerate mutase, and from 3-phosphoglycerate to phosphoglycerate is formed this position is changed to 2 position. The position is changed, so this enzyme is called mutase. In the 8th step, this 2-phosphoglycerate is converted to phosphoenol pyruvate with the enzyme enolase. When PEP is converted to 2-phosphoglycerate, this enzyme name is valid. Water molecule is getting out with the help of this magnesium ion. And the 9th step here, the phosphoenol pyruvate is converted to pyruvate with the help of ADP. ATP is generated here, 2 molecules actually, with the help of Mg2 plus, and the enzyme is pyruvate kinase. Kinase is used when is named when there is phosphorylation. So this is also for the opposite reaction, but this reaction is not reversible. And in step number 10, pyruvate is converted to lactate in anaerobic conditions with the enzyme lactate dehydrogenase. This is also named for the opposite reaction, because dehydrogenase means hydrogen is getting lost, but this is opposite from pyruvate to lactate. This should be named as pyruvate hydrogenase. This step is actually reversible, and this NADH plus this reduced form is utilized in case of anaerobic conditions, because since less oxygen is there, Krebs cycle cannot function, and this NAD plus cannot be generated from Krebs cycle. Energetics of glycolysis in aerobic and anaerobic conditions. From NADH in the step number 5, we get, there are 2 molecules, so we get 2 molecules of NADH plus H plus actually. Earlier people used to say that 3 molecules of ATPs are generated from 1 molecule of this, and now people are considering 2.5 number of molecules of ATPs, so 6 or 5 total number of ATPs from this, and 2 ATPs from step number 6 and another 2 from step number 9, and 2 ATPs are used in the first step and third step. Total number of ATPs generated are 8 or 7. In the anaerobic respiration, this NADH cannot be used because no oxygen is there. It cannot be used by the Krebs cycle. So ATPs, excluding this, we have only 2 ATPs. Now the inhibitors, iodoacetate and arsenate, these inhibits the enzyme glyceraldehyde 3-phosphate dehydrogenase, and fluoride inhibits enolase. Enolase is present in the step number 8 that converts 2-phosphoglycerate to phosphoenol pyruvate, and this is utilized in blood sugar estimation so that glucose cannot be glycolized. The significance of glycolysis: number 1, energy production in all cells. Number 2, it also occurs in anaerobic conditions. Number 3, cells like RBC and lens use only glycolysis because they lack mitochondria that has the other modes of energy formations like TCA cycle and beta oxidation. And number 4, DHAP, dihydroxyacetone phosphate. This is used to produce glycerol 3-phosphate for TAG synthesis, triacyl glycerol synthesis, and pyruvate for transamination to alanine. Glycolysis provides an intermediate 1,3-bisphosphoglycerate to produce 2,3-BPG, 2,3-bisphosphoglycerate to release. This actually reduces the affinity of oxygen towards hemoglobin and helps in dissociation of oxyhemoglobin to release oxygen to tissues. The regulation of glycolysis under the columns enzyme inducer, repressor, activator, and inhibitor. Hexokinase enzyme is inhibited by glucose 6-phosphate, and this is a stronger enzyme than glucokinase, which acts when there is little amount of glucose in the blood, but glucokinase has high is constant and less affinity. It is present only in liver and mainly for storage purpose for glycogen synthesis. Functions when there is excess glucose in the blood, and inducers for glycolysis is insulin in all the cases except this hexokinase, and repressor of glycolysis is glucagon. For phosphofructokinase, the activators are AMP, fructose 6-phosphate, and fructose 2,6-bisphosphate, and inhibitors are ATP. ATP means when there is increased in energy, this is inhibited, and citrate and less pH, more acidity. Pyruvate kinase is activated by fructose 1,6-bisphosphate and inhibited by high energy ATP. Bastyr effect is inhibitory effect of oxygen on glycolysis. Under aerobic conditions, glycolysis is inhibited. Significance of LDH, lactate dehydrogenase enzyme. Glycolysis requires uninterrupted supply of NAD+. For the enzyme glyceraldehyde 3-phosphate dehydrogenase, producing NADH plus H+, and this reduced form of NAD+ is oxidized by oxidative phosphorylation. But during anaerobic conditions, this oxidative phosphorylation is cannot work. Lactate is produced by the enzyme lactate dehydrogenase from pyruvate using NADH plus H+. So there is energy production, and this is very crucial for skeletal muscles. Lactic acidosis, a kind of metabolic acidosis, occurs when lactate concentration increases, resulting in less pH or higher acidity. It is due to overproduction or underutilization. Or the causes of lactic acidosis are pulmonary embolism, myocardial infarction, hemorrhage, shock, which leads to decrease in oxygen availability, and this leads to anaerobic glycolysis, which leads to overproduction of lactate. Physiological conditions, strenuous exercise, hypoxia, that leads to anaerobic glycolysis and leads to overproduction of lactate. Inherited pyruvate dehydrogenase and pyruvate carboxylase enzyme deficiencies leads to accumulation of pyruvate, and pyruvate is then converted to lactate. When there is a decrease in thiamine, in the dietary thiamine that is required as a coenzyme for pyruvate dehydrogenase enzyme as TPP or thiamine pyrophosphate, this leads to accumulation of pyruvate and lactic acidosis by production of lactate. Arsenite and mercury also inhibit pyruvate dehydrogenase and causes similar accumulation of pyruvate to form lactic acidosis. Then alcoholism, that decreases thiamine absorption and produces more NADH plus H+, that increases the production of lactate from pyruvate. Von Gierke's disease, that leads to decrease in production of glucose 6-phosphatase enzyme in liver, that is used for conversion of glucose from lactate. When this is defective, it causes accumulation of lactate and causes lactic acidosis. This is mainly this enzyme glucose 6-phosphatase is actually used for glycogenolysis, that converts glucose 6-phosphate to glucose in liver. This is absent in muscles. This property of this enzyme is gluconeogenic property, formation of glucose from lactate.